Okay, thank you very much. So I'm going to talk about a weird result we got recently that you'd think that when you increase the habitat, then the population, natural selection works larger in large populations, you'd be more fit. But actually, we found some circumstances in which the population becomes less fit as you increase the size of it. And this is work, the first author on it is Kevin Gomez. He has graduated his PhD, has moved into industry, and is bored enough that as a hobby, he's been slowly continuing this line of research leading up to this paper. So I say fitter and more fit. What does that even mean? So the the there are three kinds of fitness that Grime sort of categorized in what I think is a very insightful way. And so you can have fitness meaning, for example, that you are stress tolerant and you die less, and I'll call that deselected, like these brain corals here. You can also think about weediness, very good at colonizing new surfaces. That's a kind of birth or dispersal, be selected. And then you've got the competitors that overshade and crowd everyone out in a kind of zero-sum contest. That's your classic Wright Fisher model. There are always exactly in slots. Somebody's going to get every slot. You just have to be the one to try and get it. That I'll call C for contest selected. And so a project in my lab for a while has been to replace ideas of just fitness with a theory of vital rates, such as B, C, and D, that, capture these things. And one of the ways we've done this is by expanding the lottery model of ecology. So you have a certain number of slots. In each time step, a certain proportion of them survive versus die. The survivors then have the opportunity to give birth and have their propagules disperse. And then when they get to the new slot, here shown as one slot that has two of one kind and one of the other, but the kind that has more propagules and two, actually they're less than half as competitive. And so, who wins it is proportional to how many propagules land there and how competitive they are. That's the C. So we now have B, C, and D, and the cycle just continues like this. It is a generalization of the lottery model that generalizes it to densities that are not full. And the lottery model itself is a generalization of the Wright Fisher model that generalizes it to overlapping generations. So I'm not going to talk about these equations. I'm just going to say, I showed you the model, we solved it, we can write down the solution. And there isn't just a thing called fitness. In the end, what the selection is doing depends very much on density for both B and C, not for D. And, for example, if you have ecological disturbances or something like that, it'll completely change what selection is doing, as you'd expect from Grime's theory. So what I really want to focus on now is what happens when different mutations affect different traits. So eventually I'm going to get to B versus C versus D, but just to start with, if you have the two traits might also have different rates of beneficial mutations and different step sizes of how big the selective advantage for each mutation is. So stepping back just for a sec to only one trait, here's just looking at different selection coefficients and beneficial mutation rates and doing contours of parameter combinations that adapt in an asexual population at the same rate. And there are three regimes. Down the bottom, we have the origin, what I call the origin fixation regime. Gillespie originally called it the weak mutation, strong selection regime, where basically there's only one thing going on at once and it wins or it doesn't. And then there's the multiple mutations regime that was characterized by Desai and Fisher that you can see as a stochastic traveling wave with most of the stochasticity at the front and then the rest sort of moving deterministically. And then there's a really weird regime, which is sort of mutation becomes so strong that it's a diffusive force and that's kind of different. Anyway, the point is you get these shapes where two traits might have the same adaptation rate in isolation, even though they have different parameters. So the question we want to know is what happens if there are different mutations that each improve a different trait? So just say you have one trait where its selection coefficient and mutation rate are given by the dot here. And then a second trait that could have any combination in this square, how does the fact that the first trait exists change how the second trait is going to adapt? And in simulations, we found a very striking result that for traits that would evolve slower in isolation, would evolve faster in isolation, they're completely unaffected. And for traits that would evolve slower in isolation, they are completely stalled. So basically, whoever would adapt faster if the other trait weren't there carries on as normal, and the trait that would adapt slower in isolation undergoes this phenomenon called evolutionary stalling. Okay, so now let's get to the question I wanted to know. What if there's one kind of trait that is a zero-sum contest, and that in our lottery model is the C, the contest trait, and one of them that's an absolute trait that's sort of fighting against environmental change and is responsible for the absolute level of adaptation in the population. So you can do this with either B, slightly simpler results in the case of B, so I'll stick to that. D is similar. And so what you get is if you have adaptation as the B arrow, that you're getting to higher and higher B values, but at the same time, environment keeps changing you and pushing you back to lower and lower B values. And there is some point at which the two intersect, because there are diminishing returns as you get better and better to your ability to keep getting better. And so you reach that is a stable state of the system. And then meantime, just say the C adaptation is slower. C adaptation, which given B, the more adapted you are, the higher your density. C traits are more important in dense populations. So it goes up with density. But the point is here, it's not a threat. It's stalled throughout the area of interest. But an alternative scenario is that the environmental change rate is low and the rate of the relative adaptation is relatively larger. And in this case, instead of the population reaching the intersection between blue and black, it stalls at the intersection between blue and red. So as soon as blue becomes slower than red, the absolute adaptation becomes slower than relative adaptation. The relative adaptation stalls, and the environmental change sort of takes it back to the vicinity of that intersection. So now we get to the weirdness. What happens if we increase the amount of territory, which we expect to be good for a population? It's good for a population because larger territory means a larger population size and more beneficial mutations coming into it. So it's going to increase the blue, but it's also going to increase the red. And we find it depends a bit on the details of the system, but there's a variety of scenarios under which it can increase the red by more than it increases the blue. And so what happens, here's your population density, that as a result of getting all of this additional territory, the population actually ends up worse adapted. So essentially, there's so much adaptation going around that's just a brutal, pointless, zero-sum game. The real adaptation, basically the clonal interference is distracting the population from what it really needs to do to counter environmental change. And so it's worse off as a result of becoming larger, because larger populations are so much more cutthroat, and adaptation can't do anything at once and ends up doing much more of the zero-sum stuff and less of the absolute stuff. So to sum up, we do have this analytically tractable model where we have juvenile births, juvenile contests over territory in order to grow up and become adults, and then death rates when they become adults. And so we think that this way of thinking of vital rates is a better way of thinking about than sort of, naive models of what just fitness is. PopGen standard models do not fit this lottery model. they turn out to depend on assumptions that density is constant, which it's not. I didn't talk about it, but they also depend on assumptions that there's no pleiotropy between the traits. And then when each mutation only improves one trait, so there's no pleiotropy, you get this weird phenomenon of evolutionary stalling, especially in asexual populations. This is really driven by clonal interference, that the faster adaptation crowds out the slower. And paradoxically, this can mean that increasing a population size can paradoxically make the population less well adapted and have lower density because of this phenomenon of destruction. Finally, I'm more generally interested in the tension between cutthroat relative contests and absolute contests over what matters. And I wrote a book about that also in Finance and Economics. But largely, I would like to thank the people who did this work, especially Kevin Gomez, who was helped by Nathan Aviles, and Jason Bertram was the one who did the original work on the lottery model. Thank you all.
Andreas, we can't hear you. I can see your lips moving, but I think your mic might be the problem. Were you trying to say that we have a couple minutes for questions? Yes? Okay, cool. I'll see if you could sort out your mic, but in the meantime, if folks have some questions, you can either type them in the Q&A or do the raise hand feature in React, and then we can get you unmuted to chat for a moment. I know I have a question. Great talk. So thank you for sharing this work. Do you or folks in your lab have suggestions for what it might look like to begin including these considerations in these PopGen models that don't allow for this complexity that you described here?
I mean, I guess this talk to me was, the first, we're trying to do that, so this was the first step towards that. So, we began by solving the lottery model, and then this is now trying to do something with the lottery model to show how, oh, wow, there are really counterintuitive phenomena going on when you include the complexity of, yeah, there's no such thing as fitness, there's only life history traits. Andreas, I still can't hear you. But the chat should still work. Yeah, we've been sort of trying, we haven't really taken it to sexual populations yet. We have some other work that's still in review where we're sort of more just straight ecology. We're taking it to consumable resources. This is really about a territorial model. I expect these effects that I presented today to be weaker in sexual populations. This is, we're really playing up the clonal interference angle here for now. But I think it still makes the point of how different kinds of fitness are and how we should really think about how fitness interacts with density in complex ways.
It's really interesting. Thank you.
Okay, I got Andreas's question.
Yes.
The horrible question for theory, what happens if the mutations are highly pleiotropic on these traits? Yeah, so I think that's the question that more people have looked at in the past. I think that sort of the PopGen considerations of clonal interference had been less studied. I think that sort of the default as part of, say, Graham's theory was the assumption that everything is governed by pleiotropy. And I think, you can get a lot of results through clonal interference as well. So yes, sometimes things are pleiotropic. Sometimes things are just conditional mutation load dependent and there are positive correlations. Sometimes there are allocations and there are negative correlations between traits. And then when two things are adapting at once, you actually get a negative correlation just as a traveling wave that goes as a result. Anyway, next person.
Yes, we'll have our next speaker go ahead and share your screen. Thank you so much once again.
Okay, can you all see it? Okay, great.
Yes, we can see.
I'm gonna start, right?
Yes, you're all set to start. I'll take care of giving you the signals.
Okay, thank you. Okay, hi everybody, I'm Cleo, and today's talk is titled Reconstruction of the Human Amylase Locus Reveals Ancient Duplications, Setting Modern Day Variation. So this is a project I absolutely adore and I really love working on. But before I start talking about amylase and the evolution in humans, I will be talking about genetic variants. So most of the times when we think about genetic variants, we are thinking about single nucleotide variants. And although single nucleotide variants indeed make for almost 5 million bases of change between two diploid human genomes, we have all of these other types of structural variants, such as inversions, deletions, and insertions, that although the counts are much smaller than the counts of the single nucleotide variants, they make up for almost 30 megabases of differences between two diploid human genomes. And nowadays, with long-read sequencing, it's much easier to disentangle it than it was a few years back, working cytological studies, and now we have the nucleotide resolution. So a lot of the answers that remain open can now be answered. So under this slide, we're looking into the evolution of the amylase enzyme. So amylase was the first enzyme to ever be discovered in humans. It was discovered in 1931, and it's implicated with diabetes, metabolic syndrome, starch perception, and even Alzheimer's. And another interesting aspect about the amylase enzyme and the gene that codes for the enzyme amylase is that already by 1980s, a lot of things were known about the amylase gene structure. So we did know that potentially it's a copy number variable gene, which was then verified in the early 2000s. So we did have more information, but so the full locus was not resolved. So here, before I start talking about the findings, I want to preface about the amylase locus and how it looks in the human reference genome. So in the G38 human reference genome, we have 2 AMY2 genes and 3 AMY1 genes. The 2 AMY2 genes are pancreatic genes, and we have AMY2 B and AMY2A. And then we have the 3 salivary genes that they're AMY1A, AMY1B, and AMY1C, that they're almost identical between them. So from now on, I will be color coding these genes as AMY2B for the purple arrow, AMY2A will be the union of the red and orange, and the 3 AMY1 genes will be these pink arrows. So to look into the complexity of the human amylase locus, we used long-read sequencing and optical mapping for 117 haploid assemblies. So we reconstructed these 117 haploid assemblies, and we were able to identify 30 structural haplotypes. So we have all of these distinct 30 structural haplotypes that, as you can see, the primary difference between most of these haplotypes is the copy number of the AMY1 gene, so this pink arrow. So I want you to particularly pay attention in these 4 haplotypes, that they are the most common haplotypes. They make almost 70% of the distinct structural haplotypes that we have worldwide. But also notice that we have individuals or structural haplotypes that they carry a single copy of the AMY1 gene, 3 copies, 5 copies, 7 copies, and so on. So looking at these 4 more common haplotypes across the globe, we can see that we can find them everywhere, but most interestingly, we can find them in Africa, especially these 3 haplotypes, the H3 haplotypes that they carry 3 genes. They are found in Africa in very high frequencies, which suggests that probably this duplication of the amylase locus in the human amylase locus occurred before the out of Africa 50,000 years ago. Also, although we have all of this structural plasticity or all of these genomic innovations through duplications, we do observe that we have really strong negative selection, purifying selection operating in the nucleotide level. So while the genomic sequence on the structural level might be under constraint, at the same time, the nucleotide sequence is while the structural sequence is mutating really fast, we have very strong constraint in the nucleotide sequence. Also, to understand this evolution of the copy number, we looked into the oldest available anatomically modern humans. This is Pastera murae and Ustism from 34,000 years ago and 45,000 years respectively. And Pastera murae carries 8 genes per diploid cell, while Ustism carries 6 genes. So already suggesting a high copy number. Also, we looked at 64 ancient genomes that they were available to try to trace the evolution of the amylase copy number through agriculture and try to better understand these patterns. So here we are partitioning the data for particularly looking on the panel here on the right to pre-agriculture, during transition, and post-transition. And we have color-coded the hunter-gatherers in red and the agriculturists in beige. And we can see a stepwise transition. So starting from lower copy numbers, they are slightly increasing during the transition to agriculture to then higher copy numbers and also higher frequency. So these copy numbers in the post-transition, suggesting that potentially this increase in the copy number of the amylase is in response to the agriculturist diet. Also, we looked at archaic hominins. For this analysis, we used 6 archaic hominins. Here I'm showing you Goyet and Denisova. And we can see here in Goyet that we have an increase in the copy number of the AMY1 gene, potentially suggesting that the duplication is even older than we originally assumed, and it could predate the human Neanderthal split. Once again, I'm going over the structure because we have the AMY2B gene, AMY2A, and AMY1, and right now we're primarily talking about the AMY1 gene. And although we have color-coded these arrows with pink, assuming that they are all the same, there are slight differences between them. So we were able to identify these differences that separate AMY1C from AMY1B and AMY1A. This is the sequence that is flanking the gene, not the genic sequence per se. And we identified these 4 breakpoints within the sequence that they can allow us to identify and reconstruct the history of the locus. So starting from a haplotype that carries a single amylase gene that is a chimp-like haplotype, we were able to infer how humans arrived in the 3-copy haplotype. And this is quite important for what is coming next. We have a lot of PRDM9 sites in the region. So we have here looked for all of the PRDM9 sites that they could have implication in recombination in the locus, and we identified these and the degenerate ones. And now we're talking about NHR, which stands for non-allelic homologous recombination. So NHR can generate duplications, deletions, and inversions, and the model is simple. So assuming that we have 2 segmental duplications, A, B, and C, D, and we have 2 sister chromatids, the 2 segmental duplication can misalign and form this illegitimate crossover, which then would generate in a duplication where we would have a recombinant duplicon that would be CB, so crossing over here, or it could generate a deletion, and then we would have also a mosaic recombinant duplicon that copy that would be type AD. And the same, similarly, we can have inversions, but for the inversions, we don't rely on direct repeats. Instead, we require that we have inverted repeats. So here the chromosome can fold, the 2 repeats can be found now one underneath the other, and we can have a double strand breaks forming within the repeat. The sequence is excised and reinserted with a reverse orientation. So putting everything together, when we look again at the schematic representation of the structural variation in the locus, we observe that we have all of these inversions that separate the haplotypes, and they account almost for 65% of the total structural variation in the locus. So trying to understand these inversions, we looked into haplotypes that they are different between them by a single inversion, and we were able to determine that these inversions are in fact NHR inversions that occur between segmental duplications with a reverse orientation. We were able to detect the breakpoints of this sequence and also validate that these inversions are actually recurrent, so happen over and over again through the history of the locus. Another puzzling question is what happens with regards to the copy number. Like I originally prefaced the structural haplotypes by saying that we see copy numbers like 1, 3, 5, and 7. And what we observe in haploid cell is that we always have odd copy numbers, and in diploid cell we have even copy numbers. So we do have this pattern that we couldn't explain. So again, NHR is the answer that generates segmental duplications. So what we have in this case is starting by 2 haplotypes, that they're H3R1, which is the ancestral human haplotype. We can have illegitimate crossovers between these 2 orange repeats. These 2 orange repeats are flanking the two genes, AMY1A and AMY1B. And once these will cross over illegitimately, they can generate duplications or deletions. And in these duplications, we always have the duplication of the AMY1A and the AMY1B together intact as a block, or the same way the deletion of this. Therefore, we can explain the copy number of 1, 3, 5, 7, and so on. We also use parsimony informative sites that they were found between these different types of orange repeats, and we were able to observe the occurrence of these chimeric AB and BA-like sequences that confirm that NHR is actually occurring and is occurring between these sequences. Yet again, this is not the only mechanism that generates breaks and generates duplications. We also have an MMBR mechanism, which stands for microhomology-mediated break-induced replication mechanism, which, unlike NHR, is not a recombination-based mechanism. Instead, it's a replication-based mechanism. And again, starting from a haplotype that is H3R1, like the ancestral haplotype, we would have to infer that the recombination fork would stall, and then it would jump to another active replication fork and be rescued by microhomologies and generate novel haplotypes. We were able to determine these microhomologies at the breakpoint junctions, and also we identified in total 4 structural haplotypes that they are the result of MMBR, identifying all of the microhomologies. And what we observe is that they mostly seem to influence the AMY2B and AMY2A genes rather than the AMY1 genes like with NHR. So putting all of this data together, we were able to reconstruct this mutational network where we can connect all of these 30 distinct structural haplotypes to one another, inferring one single step, like a single inversion or a duplication event, or sometimes multiple events, so we can explain all of the present-day structural variation, starting from these 4 haplotypes that they seem to be the most common worldwide. So given all of these evidence and all of the findings together, I think that summarizing, we can say that potentially the first duplication in the amylase locus could predate the human Neanderthal split. We already observe at least 6 copies of amylase genes per diploid cell in Ustissem 45,000 years ago, and we observe a stepwise increase from pre-agriculture to post-agriculture with an expansion in the copy number of the amylase gene to the present-day data, where we can observe this population of variable substrates with all of these duplications, deletions, and inversions happening at the same time. And with this, I would like to thank everybody that participated in this work from the Gokshuman Lab and the Lee Lab. I want to thank the funding bodies, that is NIH and NSF, my colleagues here that we are putting their amylase to use for me, and the organizers for giving me the chance to talk. And if you want to check the manuscript, you can scan the QR code. Thank you very much. And I'm very happy to take any questions.
All right, let's give us a shot again. Can you hear me?
It's working.
Great talk, Cleo. Perfect.
Thank you.
As we should have a couple minutes for questions, so please drop them either in the Q&A or raise your hand. I guess I'll hop in with one of them. You showed in one of those last slides a little bit about the potential timing of these evolutionary events. But you have this really deep network, a pretty cool resolution of all the potential different mutations that were involved. Is there any strategy to be able to time all of those different steps?
It's a little bit hard because a lot of these are recurrent. So I will show you something that for simplicity I didn't include. So this is the phylogenetic network. So you can see, and also an LD plot. So you can see that we have really, really weak LD. So a lot of these structural haplotypes are regenerated over and over again. And in fact, I've been working with a lot of trios to try to delineate like a single trio that we have a novel structural haplotype occurring or a novel inversion. And there are a few, but it would be very hard because for the MMBR events, we know that these are single origin events, so they occur once and we can trace them back when they happened, and they are nested together very nicely in the tree. But for the NHR events, I think they are so recurrent that if you look at the H1 haplotype, which is a different H1 haplotype than the ancestral H1 haplotype, is the deleted H1, we can find it everywhere through the tree. And also, like with PCAs, we do get similar results. So I think that the primary duplication we can pinpoint, but the secondary duplications will be very hard. And a lot of the H3 haplotypes are also secondary rather than primary.
Awesome. Well, thanks so much. You can stop sharing and let's have David start up his slides. We have just about 30 seconds. All right, and we'll wait for you to start right on the dot so we can stay on track.
Okay.
All right, my clock says 6:30. Off you go.
Hi, everyone, and thank you. My name is David, and today I'll be discussing a theoretical model and an interactive app that we developed to explore how chromosomal inversions might couple with ecological and mating bias multi-locus genotypes to promote reproductive isolation in Sympatric speciation. And so our group is looking to develop a complete mechanism to show how Sympatric speciation can occur. And one of the questions we'd like to answer is how can we overcome the homogenizing effects of gene flow? And so since Sympatric speciation relies on like mechanisms that promote differential assortment of adaptive traits, it's important to understand how these mechanisms, what kind of mechanisms can play a role in that. And so one of those mechanisms we believe is that can aid in this is chromosomal inversions. And we think they'd act as a late stage adaptive barrier mechanism. We say late stage because there needs to be some non-random assortment of advantageous alleles already present in a population. This would make it easier for a chromosomal inversion to capture these advantageous combinations, and adaptive in the sense that they need to preserve the beneficial allele combinations. That's only really advantageous if hybrids are less fit. So without this hybrid disadvantage, the chromosomal inversion really has no benefit and wouldn't be able to invade. So we believe the chromosomal inversions can form supergenes that will reduce recombination and maintain the linkage between these adaptive traits. So this work that we've done, it builds off of a previous model. So in our previous model, we had a 3 ecological locus model with 1 mating bias locus, and it did not allow for viable hybrids. In this model, since most phenotypic traits are polygenic in nature, we wanted to understand the dynamics of these polygenetic traits, how the dynamic of these polygenic traits are assorted is like integral to the process of Sympatric speciation. So yeah, we allow for multiple ecological loci and multiple mating bias loci. And we also introduced viable hybrids into our model, and then chromosomal inversions can capture any combination of ecological or mating bias alleles. So we have a picture of the general algorithm that was developed by Dr. Lin and Curtis. And then on the right, we have our mathematical model that was developed by Curtis. Okay, so I'll go ahead and start with the ecological model. So the way the simulation works is each individual will have multiple locus ecological genotypes, and they'll have either an A or a B allele. And then the ecological genotype will determine which niche each individual is assigned to. And then the individuals in the ecological model carry 1 mating bias allele, either an X or a Y value. And then the mating success is determined by the mating bias allele. So if they have the same mating bias allele, they'll always mate 100% of the time. If not, then there's a parameter we developed called alpha that'll determine the mating bias success. So they'll go through a number of matching rounds, and after a certain number of generations, we can introduce a chromosomal inversion into the system and then proceed again with the simulation to see how the chromosomal inversion affects reproductive isolation. Okay, and so this is a way that the app interface looks. So main takeaway here is on the left-hand side, we have a face portrait, and what we're looking for is strong reproductive isolation. And that'll be determined by the face portrait, the fixed point of the face portrait converging in the bottom right-hand corner and the top left-hand corner. That determines the number of pure genotypes and what percentage of them have the different mating bias allele. With this app, we're also able to track the rate that the chromosomal inversion would invade a population, which is shown here by this line graph. And then we can keep track of the distributions, genotype distribution for the population at the beginning, before the chromosomal inversion invades, and after the chromosomal inversion invades. Okay, and so going on to some of the results from the model. So first we wanted to see what happens when we increase the number of ecological loci before introducing viable hybrids. And this confirmed a previous finding that we had, which was as you increase the number of ecological loci, it strengthens reproductive isolation. That's due to the fact that in this case, hybrids would still be considered maladaptive. And so as you increase the number of loci, you have more hybrids that are produced. So they are eliminated due to the ecological disruptive selection. And as that continues, the disruptive selection intensifies and leads to stronger reproductive isolation. So what we have here is two simulations. The one on the left is a two loci simulation. And you can see that the fixed point shifts towards the bottom right-hand corner, signifying stronger reproductive isolation once we go to a 4 loci system, with all other parameters being held constant. Okay, the next result that we found from the model was what happens when we introduce the presence of viable hybrids. And what we found was that this weakens reproductive isolation, which was expected due to the increase in gene flow. And again, the weaker reproductive isolation is shown by the shift away from the corners of the fixed point when we go from a system that does not allow for viable hybrids to one that does allow for viable hybrids. So another key finding was what happens when a chromosomal inversion only captures the ecological alleles. And what we have found is that negative coupling occurs. So the chromosomal inversion is able to quickly invade the system due to a fitness advantage, but that ends up disrupting the ecological selection process needed to maintain reproductive isolation. And so as these fewer hybrids are produced within the niches, sexual selection ends up undermining the ecological divergence. And what we can see here is that the system seems to converge on one of the mating bias alleles. So in this case, we have a system that captures all of the ecologically adaptive alleles. So it captures the AAA allele, and this green dot here signifies the start before the chromosomal inversion invades. And as we move towards the diagonal, we see that we lose the X mating bias allele, and it's also representative here in the bar graph. So at the beginning, the A niche has a fair amount of the X allele, but as the chromosomal inversion continues to invade, it converges on the Y allele. Yeah. Next, we wanted to see what happens when we are able to pair a chromosomal inversion that captures both an ecological and a mating bias allele. What we found is in this case, it can strengthen reproductive isolation. So this is due to the fact that the mating bias sort of has a hitchhiking effect. And so now the chromosomal inversion has a shared selective advantage where it can invade different ecological niches, and it also has paired with the mating bias allele. So what we also found was that as the chromosomal inversion is able to capture more ecological alleles, it also leads to a faster invasion rate, which is shown here on the right-hand side. Although when just one ecological allele is captured with the mating bias allele, it leads to complete fixation. So again, we have the green dot before the chromosome inversion in the single line phase portrait, and it reaches all the way to the bottom right-hand corner. When you have more than one captured, it leads to almost complete fixation, but nonetheless stronger reproductive isolation. Next, we'll move on to the mating bias model. So in this model, each individual has multiple mating bias loci, and they can take a value of X or Y. In this model, each individual belongs to one of two niches, either A or B, and the individuals will randomly encounter one another, and their mating bias is determined by a mating bias matrix. So if they have the same genotype, they'll mate again 100% of the time. If they have a different genotype, then their mating success decreases linearly away from the diagonal, depending on how similar their genotype is. Yeah, and so they will go through a number of matching rounds, a number of generations, and then again, we can introduce a chromosomal inversion and see how the system progresses after the chromosomal inversion is introduced to the system. Again, really quick, this is just another picture of the app interface for the mating bias model. Again, we're looking for strong reproductive isolation. So we want the system to converge on the corners, the top left and the bottom right-hand corner. It's a little hard to see in this graph, but you can see the effect that the chromosomal inversion has on a system. It moves from this like green dot to the red dot in the all lines form of the face portrait. And then this graph here, the one in the middle, shows the invasion rate of the chromosomal inversion. So in this model, a chromosomal inversion can be introduced to either of the two niches. And then again, we have a bar plot that shows the distribution of the genotypes. In this case, the blue represents the individuals that belong to the A niche. The red represents the individuals that belong to the B niche. So moving on to the results for our multi-locus system. So what we found was that as you increase the number of mating bias loci, you end up having a more viable hybrid production. And as you increase the hybrid genotypes, this reduces the reproductive isolation. So in a sympatric population, what we would want is fewer mating bias loci. And yeah, when we have a chromosomal inversion introduced into the system, what we found is as it captures more high mating bias alleles, it'll have a stronger fitness advantage. And the stronger fitness advantage leads to effectively reduces the number of loci, since the chromosomal inversion is suppressing the effects of recombination. And the fewer hybrids will then lead to stronger pre-mating reproductive isolation. So one way we showed that here is we have two bar graphs from two different simulations. The one at the top is where 2 of 3 mating bias loci are captured, and the one in the bottom is where all mating bias loci are captured. So you can see that the one in the bottom, all of the hybrids end up being eliminated. The light blue and the light pink mean that these are individuals that have the inverted regions. The dark red, the dark blue are the individuals that have not undergone that don't contain an inverted region. Yeah. So to summarize our model, yeah, we developed a model that can factor in any number of ecological or mating bias loci. We ended up confirming previous findings that increasing the number of ecological loci or decreasing the number of mating bias loci facilitates the emergence of pre-mating reproductive isolation. Presence of viable hybrids gives rise to chromosomal polymorphism. That was something I didn't touch on. I think I was like rushing through. But when we have these hybrids and they're not affected by the chromosomal inversion, they continue to resupply the hybrid niches. And that way, so in the ecological model, you can't really have complete elimination of those hybrid niches. But yeah, the chromosomal inversion is also able to capture the ones that only capture ecological alleles, leads to a decrease in reproductive isolation through negative coupling. When a chromosomal inversion is paired with an ecological and mating bias allele, we have stronger reproductive isolation through positive coupling. And a chromosomal inversion that captures high mating bias alleles leads to fewer hybrid genotypes and stronger reproductive isolation. Some of the limitations of our model is that it assumes all loci are fully linked. It doesn't model crossover events. There are only 2 niches and binary alleles systems, and we would need some empirical validation in order to validate the inversion dynamics and the hybrid structures. So yeah, I want to thank everyone in our lab and everyone who collaborated on this project. And with that, I can open it up for any questions. Thank you for your time.
For that talk, David. I think we have time just for one really quick question. If anyone has it. All right, I'll jump in again. I love inversions. I love thinking about mating preference and inversions. Have you tried simulating something that's a more classic preference trait model where one sex expresses a preference, the other one has the mating trait, right? So different loci are affecting both the preference and the trait. And there's a match between them. Right now you have a multi-locus thing where there's just a match between the total sum of traits, but they're not separate sets of loci. That wondering if you've taken a look at anything like that.
No, I don't. I'm not sure if we have. We have a couple of people from our group that might have been working on something similar to that. Yeah, I don't think we've looked into that. That's a good point. I can go back to my PI and we can see if we can factor it into the model later. Yeah, thank you for the question.
Stop sharing. That's just about hands up. Off on. Go ahead, Karyn.
Okay, just wanted to make sure. Okay. So let me pop this up. And then, are you able to see everything okay? Just want to confirm.
You're all set.
You can full screen it. Right now I'm seeing it in the center of your screen, but we
It's
can
not
see
showing the, are you, is it, it's not showing the full screen? How about now?
perfect.
Okay. All right. So I'm going to be presenting today on specifically coupling of habitat preference barriers and how it leads to reproductive isolation in sympatric speciation. So the question we mostly focused on was, what role does habitat preference play in initiating pre-mating barriers and coupling with other barrier mechanisms to establish robust reproductive isolation in sympatric speciation? So to give you guys a brief introduction on how we began this project, we investigated how habitat preference barriers in two spatial configurations based on existing examples of sympatric speciation. So the first one, the open space system, is based on a population of cichlid fish, which underwent sympatric speciation in the open lake. And our second one, the no open space system, is configured based on the hawthorn and apple maggot flies example that speciated over time. And to give you some more background on what an open space and no open space system in our configuration means. So here we have a mathematical model that we created on the open space and no open space systems. So for the open space model, AH represents the homebound individuals that remain and mate within niche A, and likewise for BH as well. So for the AF and BF, which are the free roaming individuals from their respective niches, they're able to swim out into the open space, and which is represented by this red line, and mate. So notice that they can also mate with their native niches, but cannot swim into the opposing niche. And the no open space model shows AH and BH as the respective niches, and FF represents the free roaming individuals from both niche A and niche B, as they are able to move out to each niche. So in the no open space model, the mating occurs only at niche A or niche B, not in between. So what we found is that from the no open space system, it allows for more free movement and is still able to lead to faster and stronger reproductive isolation in compared to the other model. And in the no open space model, this is partially why the reproductive isolation is a little weaker, is because of the homebound individuals that are still being able to be visited by the free roaming individuals from each niche. So onto our methodology on how we designed all of this. So our habitat preference model, which is shown here, is broken down into more specific mathematical models, which were used to simulate populations in which we introduced the habitat preference barrier and examined for reproductive isolation. So we used our study used MATLAB and its app designer tool to generate user-friendly graphical user interfaces that solve the mathematical models of habitat preference barriers and their coupling with the 2 allele mating bias barrier from a previous study. If you're interested in learning more about what our mathematical models are and how the computer simulation started with the programming, here's a QR code linking to a previous video that our team has presented in the past. So here are some key takeaways that I'm going to discuss that we discovered from results in regards to habitat preference establishment, 1 allele versus 2 allele, coupling facilitation, and invasion resistance pattern. So for our first point, we found that disruptive ecological selection and maladaptive hybrid loss are needed for habitat preference establishment. What this means is, so if we look at our key variables, the offspring return ratio F, which represents the strength of the disruptive ecological selection and determines the extent of how much hybrids we lose, which is also represented by HR, is available to which is needed to drive the invasion and the coupling of habitat preference barriers. And our second variable theta represents the strength of the barrier, the habitat preference barrier, and it specifically specifies how effectively it reduces maladaptive hybrid loss. So to break down this phase portrait even more, the blue dots are the starting generation and the red dots are where the generation ends up. So in this case, we ran 200 generations in here. And the diagonal line represents that the ratio of alleles in this case is equal in both niches. So because there's no maladaptive hybrid loss represented by the hybrid ratio, which equals 0, habitat preference barrier in this case is unable to invade and is unable to establish reproductive isolation. And then to move on to give you some background on what a 1 allele versus 2 allele model is. A 1 allele model focuses on a single mutant allele that produces the same effect across different niches. So we only introduce 1 allele. So when the hybrid ratio is greater than 0, the 1 allele habitat preference barrier is able to invade and move towards fixation, and it's unaffected by recombination that we found which can be caused by gene flow. So here's a phase portrait of what a 1 allele model looks like. So as you can see, these starting generations are all moving towards a fixed point established by this 1 allele. And in the 2 allele model, there's 2 different alleles that exert effects on different ecotypes. So the 2 allele model tends to converge towards a fixed point polymorphism, which is more difficult to establish due to recombination. So that means what polymorphism means in this case is that the proportion of the functional alleles exerting the habitat preference effect. So we're introducing 2 habitat preference alleles, and its effects on each niche is interacting with each other, which makes the overall effect lower. So everyone in niche A has a K allele habitat preference, which is impacting them. And for B, it would be the J allele. So as a result, what we found is that from 1 allele is easier to evolve and produces a stronger reproductive isolation just because it moves towards fixation, that polymorphism. And our third point is that we found that habitat preference can facilitate the coupling of mating bias barriers. So if we look at this phase portrait, the initial generations are moving towards the center line. And this is a current 2 allele mating bias barrier that is divergent, meaning that it's unable to produce a fixed point polymorphism and unable to produce a pre-mating reproductive isolation. However, if we reduce the theta from 0.9 to 0.6, which means strengthening the habitat preference barrier, we're able to move towards a coupling polymorphism on both sides, which means that it's establishing reproductive isolation because the habitat preference barrier is limiting the gene flow and facilitates the coupling of the mating bias barrier and strengthens the overall reproductive isolation. This is mainly accomplished by suppressing the homogenizing effect of gene flow, which would otherwise inhibit the non-random assortment of mating bias, not allowing it to couple. So to move on more about that point, so we just discussed the mechanism, which is we are able to couple mating bias and habitat preference. But what happens if you further restrict the gene flow? We notice that an invasion resistance pattern will appear as we try to strengthen the restriction. So as we decreased a number, so from earlier, we had this one where it was a divergence and it converged into a two single polymorphism where it's able to establish reproductive isolation. But as we decrease the number of matching rounds, so from 10 to 4, it creates the emergence of an invasion resistance pattern. So our AX mutant mating bias allele right here, 0.01, is unable to invade into the system and moves towards 0, meaning that it can't establish into the system. So you might wonder, why does this happen? How shouldn't it help? So we notice that there's a balance between a mating bias alleles, fitness advantage within inter-niche mating, so mating between niches, and its fitness disadvantage with intra-niche mating, which is caused by sexual selection. As a result, so intra-niche sexual selection is more likely to eliminate smaller populations of high mating bias mutants, which is why we end up finding a resistance pattern. So to combat that, we found that invasion resistance patterns can be usually eliminated by increasing the number of mating rounds and decreasing the number of niche sites. So since we just went over a mating round, I decided best way to show is the niche size. So here was earlier what we have from the invasion resistance pattern. So if we decrease the niche size from 0.6 to 0.1, there's like a slight little zone here that shows up, where there's a space where the mutant bias allele is able to invade. And as it's shown, so we increase the mutant bias allele to AX equals 0.2, which is able to then invade and actually move towards a fixed point polymorphism. So it was actually able to establish a pre-mating reproductive isolation, whereas it was initially blocked by doing so before we decreased the niche size. So to move on to our coupling data, as we tested the amount, if we could couple amongst multiple different barrier models. If you would like to know more about what our specific barrier models mean, here's a link to, here's a QR code to the program list and descriptions if you would like to look over what each of these barrier models are described as. But we wanted to check if we noticed that one specific one couldn't couple. So why didn't this one couple specifically? If you notice here, there's a hybrid ratio that equals 0. So as long as there's no hybrid loss, the habitat preference was never able to establish in the first place. So it wasn't able to facilitate any coupling. So there was no, after these generations, there was no barrier being established. So you might wonder, does coupling order actually matter then? Because earlier when we discussed everything, we had habitat preference established first in the system, and then mating bias established second. And as we discussed earlier, once habitat preference is able to invade into the system, it's very specific. Mating bias barriers are very specific and couple under certain conditions. So it's able to couple, no problem, but it has to fulfill the requirements for mating bias to be able to couple in. Whereas we tried also coupling mating bias first and then introducing habitat preference. And although mating bias was initially more difficult to establish into the system, however, whenever we established mating bias first, habitat preference was always able to couple. So in summary, habitat preference is easier to establish and tends to actually produce a stronger reproductive isolation in open space versus no open space. And as long as we've found that there's maladaptive hybrid loss, the habitat preference barrier is always able to invade to reduce the hybrid loss to the extent allowed. And a one allele model was able to generate stronger pre-mating reproductive isolation, mostly because it wasn't affected by recombination, and actually leaves a strong compared to the two allele model, which heads towards polymorphism, it heads towards fixation. And for our fourth point, so when habitat preference barrier is our first barrier mechanism, it is able to facilitate coupling of the subsequent mating bias barrier by decreasing the inter-niche encounters and mitigating the homogenizing effects of gene flow, which prevents the actual assortment of mating bias alleles. And on to our last point, we noticed that because it is so easy to evolve, the habitat preference barrier is ideally suited to establish as the first pre-mating barrier because it's actually able to encourage coupling of mating bias barrier easier to introduce and produce better reproductive isolation. And here is a picture of all our lab members and collaborators. Some of my lab members are presenting today, and you'll be looking forward to some of the other presentations. And if you have any questions, please let me know. Our preprint is actually coming out and it's going to be uploaded to arXiv in a month. So if you have any questions about it and you want to look at more data about what we did, you can look on that. And I'm ready for any questions if anyone has any.
Thanks so much for that talk, and we do have time for some questions.
Oh, yes.
Yeah.
For Sarah.
Hi, great talk. Super interesting. It is a little outside of the scope, but I'm curious if your lab group has talked about this. So I think this is really great work on putting together the order of various prezygotic barriers when it comes to ecological speciation. And have you considered how some of these models and some of these techniques you're using might be applied to understanding what prezygotic barriers get reinforced first upon secondary contact?
We have been testing out like secondary, so like David earlier with his chromosomal inversions. But right now we've been testing out mostly how habitat preference barriers and mating bias barriers interact with each other. And we've noticed that habitat preference barriers are easier to establish. So it's been able to facilitate a little better than mating bias. So we've been testing out to see how mating bias is working. but I think we had like a recent finding that we're still investigating about specifically how mating bias could possibly strengthen it. So we're still in kind of like in like a investigative like testing out other types of like pre and post barriers that could possibly impact how like sympatric speciation and reproductive isolation.
All right, you can stop sharing. We can have Lauren set up next dot. Thanks so much again. And feel free to start, Lauren.
Sorry, 1 second. Okay. All right, there we go. Sorry about that. So today I will be presenting on one of our papers entitled Computer Simulations and Analysis of Coupling Among Reproductive Barriers in Late-Stage Sympatric Speciation. My name is Lauren, by the way, and a couple of my lab partners, Karyn and David, also presented earlier, so some of this information may be familiar, or some of the modeling might be familiar, but let's get started. So in a previous study, we developed a mathematical model and derived this 2 mating bias allele to ecological niche model from this previous study. We also used individual-based computer simulations to propose this 5-stage process for establishing initial pre-mating reproductive isolation in sympatric speciation. So the aim of this study is to take the same model and these same computer simulations to investigate coupling of additional reproductive barriers during later stages of speciation. Some important things to note here, just some variables that are going to continue to come up. The variable alpha here changes mating biases and affects pre-mating reproductive isolation between ecotypes, whereas variable F changes hybrid viability and post-mating reproductive isolation. So in this last diagram, figure M1 at the bottom shows the life cycle of a sympatric population used in our computer simulations. So in each generation, individuals in the population undergo ecological selection and then sexual selection to produce offspring for the next generation. Here's an example of the computer model we developed to solve the set of nonlinear difference equations from our mathematical models. It should look familiar from a couple of the previous presentations, but you can see all of the relevant variables here. For example, that variable F and alpha mentioned earlier. All of the parameters here are normalized, and in the phase portrait, you can see we plot AX versus BX, AX being the ratio of individuals in niche A with the X allele, and same for niche B. So as we change the parameters here, we can see how the phase portrait changes. The blue dots are the initial starting populations, and here after 100 generations, you can see how the population ratios of AX and BX converge to a fixed point polymorphism shown by the red dot. So as they move closer to the bottom right hand and top left corner of the graph. So getting more into this paper, this diagram really represents the central focus of our paper and what our results showed. We analyzed 5 barrier mechanisms, 2 in this early stage, so mating bias barriers and habitat preference barriers, and 3 designated in late stage barrier mechanisms. So that's chromosomal inversions, mutations reducing hybrid viability, and BDM incompatibilities. So what we found was that these 5 barrier mechanisms work in a positive feedback loop, which I'll definitely get to later. But first, we're going to look at how these mechanisms act individually. Starting with the early stage mechanisms. So as I mentioned, the 2 early stage mechanisms will be habitat preference and mating bias barriers. And these, both of these barrier mechanisms we found to be adaptive, so driven by their selective pressures created by hybrid loss. Both of these mating bias and habitat preference barriers can independently establish initial barriers because of this, which can then couple with other similar adaptive pre-mating barrier mechanisms to progressively increase pre-mating reproductive isolation and further reduce gene flow. As mentioned earlier by Karyn, because habitat preference barriers are more readily established than the two allele mating bias barrier, establishing habitat preference first and then coupling it with mating bias, it presents a possible pathway for initiating pre-mating reproductive isolation in sympatric speciation. But also something to note is both of these are much more easily reversible. So let's go ahead and look at some of the results. So we ran these computer simulations across a bunch of different combinations of these barrier mechanisms. So these are just a couple of examples. This first one is coupling of initial two allele pre-mating barrier mechanism with a one allele barrier mechanism. And the second one is a two allele pre-mating barrier with another two allele barrier mechanism. And so in these diagrams, you can see again that the red dot moves closer to the bottom right-hand corner as the fixed point polymorphism moves towards fixation in the niches, representing stronger pre-mating reproductive isolation. So kind of coming back to this, we're going to dive into the late stage barrier mechanisms, and those are a little more complex, so I'm going to spend a little bit more time on them. These are our 3 late-stage barrier mechanisms that we analyzed. They are adaptive and non-adaptive, depending on which one we're talking about, and we'll get to that. They do occur in pre and post-mating, affect pre and post-mating reproductive isolation, as opposed to the early stage mechanisms, which only affect pre-mating reproductive isolation, and they are less easily reversed than the early stages. So first, mutations reducing hybrid viability. So these mutations are late stage as they are underdominant, so they're affected by gene flow. In the early stage, you have a lot of interniche mating and increased gene flow, and in the late stage, you have less gene flow, which causes this a smaller fitness disadvantage for these mutations reducing hybrid viability. They are adaptive as your fitness advantage is controlled by gene flow. For example, a mutant allele that increases hybrid incompatibility and decreases hybrid viability can't invade by itself, has a difficult time invading by itself because individuals carrying the allele are at a fitness disadvantage. However, when coupled with an allele that has an invasion advantage, whether it's through physical linkage or chromosomal inversion, the mutant allele can hitchhike with the advantageous allele and invade. So this can combine with chromosomal inversions and or BDM incompatibilities in order to gain this net fitness advantage necessary to invade. After the mutant allele is able to invade and rise to near fixation, even if disruptive ecological selection is reduced, the intrinsic hybrid incompatibility conveyed by the mutant alleles can still prevail to ensure continued hybrid loss and maintain that established fixed point polymorphism and pre-mating reproductive isolation from earlier so that the pre-mating mechanisms don't collapse. Moving on to the next one we're going to look at is chromosomal inversions. I'll keep really brief because we did see a presentation on this earlier from David, but chromosomal inversions are important as they're another late stage barrier mechanisms. They are dependent on non-random assortment. So when you have non-random assortment, obviously reduced gene flow, it's easier to capture favorable alleles, and they are adaptive. So these linked alleles, or we call them super gene in our paper, they can't be broken up, so they're advantageous compared to other individuals to invade. There is a gene flow intermediate range, so gene flow too high, you can't capture locally adaptive alleles, and when the gene flow is too low, there's not enough hybrid loss. We do have this issue of negative coupling, where if you capture only ecological adaptive alleles, this decreases ecological selection against hybrids necessary for pre-mating reproductive isolation. So you do need to capture both ecological and mating bias alleles in order to prevent this negative coupling. And here I just included another example from our results section of a two allele pre-mating barrier with chromosomal inversion. All right, moving on to the last one is going to be our BDM incompatibilities. So this is a late stage mechanism It operates best under low gene flow. It is non-adaptive. So as opposed to the first two late-stage mechanisms that I talked about, BDM is not driven by hybrid loss. So it's non-adaptive. It's an incidental byproduct of ecological adaptations. It is the slowest mechanism of all of the ones that we looked at. So it evolves the slowest, but it is the most irreversible. So in the final stages of reproductive isolation, when gene flow is very low, BDM incompatibilities we found can evolve independently as the genome continues to diverge, and makes this very difficult to reverse because they're all independent of each other. And here's another example of from our results section of coupling an initial 2-allele pre-mating barrier with a 1-gene locus BDM barrier mechanism. This I just included. It's part of our results section as well. It's in our paper. If I'll have a link, a QR code to it at the end. But this kind of discusses in more terms what I went over and the differences we found between how these barrier mechanisms act in early stage and late stage, and also the similarities. So finally, I did mention briefly at the beginning this positive feedback loop. So now that we've kind of dived into how the differences between these barrier mechanisms, we looked at how they all interacted with each other. And what we found was that the early stage mechanisms establish initial pre-mating isolation barriers, so this initial pre-mating reproductive isolation, and those barriers kind of chokehold and reduce gene flow, which facilitates late stage barriers that are dependent on that reduced gene flow. Then the late stage barriers can strengthen, produce post-zygotic reproductive isolation, and further reduce gene flow. And then they also secure those early stage mechanisms by with intrinsic hybrid loss to sustain pre-mating reproductive isolation, even if disruptive ecological selection does decrease. The order that barriers are coupled does matter because coupling barriers becomes more difficult as the reproductive isolation strengthens. And as I mentioned with the BDM incompatibilities, at the very last stages when there's very little gene flow, many different post-zygotic mutations and barriers can evolve independently of each other, making it difficult to reverse. So in conclusion, we extended a previous mathematical model to demonstrate how an initial barrier can facilitate coupling of additional barrier mechanisms. We used computer simulations and analyses to investigate coupling among different reproductive barriers in Sympatric speciation. Our results showed that after initial mating bias barriers reduce gene flow, it can facilitate the coupling of other barriers to produce stronger and irreversible reproductive isolation. And we saw that this works in that positive feedback loop that I kept bringing up between the early and late stage mechanisms until you have stronger and stronger reproductive isolation. And so together the results of our two studies, that first one that we keep mentioning with where we developed this mathematical model, and the this study now where we're kind of looking at the overall possible combinations of different barrier mechanisms, outline a possible pathway forward for the progression of Sympatric speciation. While our proposed mechanism still requires empirical validation and more theoretical refinement, we are just trying to offer a valuable framework that could be improved and extended through future research and discussions to advance our understanding of Sympatric speciation. And with that, there we go. Oh, sorry. This is the QR code to our preprint, excuse me, if you have any questions at all. There's obviously a lot more information in there, and I was kind of trying to move through it as quickly as possible. And I want to thank all of our lab members and collaborators and take any questions at this time. Yeah, or you can contact us with any questions as well. But thank you.
A few questions.
Andreas, you might need to repeat that. You cut out at the beginning and then it came back.
Thanks again, Lauren. We do have time for some questions. I guess because no one's asking, I'll ask. So there's plenty of empirical examples where we know that BDMs evolve quite quickly. And we also know that in some cases they are actually quite reversible. How does that fit into your framework?
Yeah, so within our framework, we were specifically looking, I think within like the certain parameters that we set up for the BDM incompatibilities. And we found that in combination with the other barrier mechanisms that they evolved much slower than say like the mating bias barriers or even the chromosomal inversions, and that they, I think more when you're looking at the combination of all of what the BDM incompatibilities can open up, then you start to look at kind of as they work in this positive feedback loop that they can make it less reversible, kind of at the end, like as a final stage, I guess, in our process specifically. But yeah, that's a really good question, and I know that we still have a lot more, I guess, like running of simulations to do, and I would be interested to see if we kind of change the parameters to better fit that that question. Yeah.
Gotta move on to the next talk. So
Thank you.
And Gregory.
Do we have time for another question?
We do not, sadly. Please post in the Q&A, or yeah, they should be able to answer it in there.